Good day, KIF1A community. It's my pleasure to welcome you to this session. My name is Amit Rakhit. I am the President and Chief Medical Officer at Ovid Therapeutics, and I'm here just to say a few words of welcome. I have a few slides to share, so I'm just going to change to slide view, and hopefully you'll all be able to see this. Beginning. And I'll put myself here. So, well, it's my pleasure to be here with you today. Just a few words on behalf of Ovid Therapeutics. We are a rare disease company that's based in New York City and focused on rare neurological conditions, and so conditions that affect the brain. And we hope to transform medical practice by creating new therapies that affect and can really make a difference in the lives of people living with serious illnesses in neurology. And part of the reason we're here is because of you. We've followed you for many years and have seen the community grow. And here's just a picture that we know of you working hand in hand with us over the last few years, and have been just thrilled to see the progress that has been made with the potential new therapies in this space and the research that's advancing every day. Well, a little bit about how our approach to creating new medicines and therapies. We really want to focus on people who are living with these serious illnesses. That means you're the people who are affected, maybe your children, your family members, and the families themselves. We understand what matters, and what we do. We take quite a bit of time to understand what is it like living day to day with a condition like KIF1A, and what does it take to really affect and make a meaningful impact in a person's life that has KIF1A. And we incorporate that understanding into our development plans very early in the research progress, very early in our development plans, very early in incorporating across the spectrum of what it takes to create a new therapy. Along with working with you and family members and the community, we work with the scientific community, raising awareness for people who don't have maybe not heard of these conditions like KIF1A, but also working with collaborating and with researchers, just trying to advance the science. And the rare disease community is very important to us because we know there are more than 7,000 rare conditions out there, and many of them have very limited therapies or no therapies at all that have been approved. And it's working together with advocacy organizations to really address this unmet need, this urgency to find new therapies and really accelerate research. And that's what's really driving much of what we do every day. Together with our communities, we accelerate R&D, the research and development process. We develop medicines based on our understanding of the science of the key biological pathways and the central role they have in neurological conditions. And then we take that understanding and really develop clinical programs that are really meant to understand criteria that are really important for you in the real world. So what are the benefits? The benefits that we're looking for are what's impactful for you in the real world, whether it's being able to have more motor coordination, whether it's speech, whether it's sleep. We want to understand what's really being impactful or will be impactful for you and clinically meaningful. And those are the criteria or endpoints that we typically will want to incorporate into our development programs. And we do this with a focused effort on the unmet need. And really, we will focus on a single indication first, and maybe that knowledge will enable us to address other conditions that are similar. And then we can approach these conditions from a science perspective, from a patient-focused perspective, from a family perspective, and really hope to make a meaningful difference in the treatment practice and create new medicines along the way. So we're very excited to be here. We're very excited to be part of the KIF1A community. Our collaborations with Dr. Wendy Chung at Columbia have really opened our kind of sense of urgency and driven us to really think about what's next in this space. And we hope really we can work together to make a meaningful difference. So thank you from all of us here at Ovid Therapeutics for allowing us to be a part of your journey. And let's continue this journey together to really find new therapies that will make a difference for all of us in this community. Thank you so much. And we look forward to working with you soon.
Hi, everybody. My name is Patrick Sarmiere. I'm the Senior Director of Molecular Pharmacology at Ovid Therapeutics. And today I wanted to talk to you a little bit about our program, OV815, which is our program to develop novel therapeutics for KAND, or KIF1A-associated neurological disorder. We've been working with Wendy Chung at Columbia University for the last year or so and coming up with approaches and actually doing some of our initial screens to identify these candidates. So I think it's an exciting time. I appreciate all what you're doing, your families, and what you're doing to support these programs. It's an exciting time, like I said, and I'm glad to be here speaking with you. Just to give you a quick background, I think most of this audience will not need to hear this, but still for continuity, we'll just go through it really quickly here. But as you know, as many of you know, that KIF1A is primarily an autosomal dominant gain-of-function disorder, where missense mutations or single nucleotide changes in the genetic code for KIF1A lead to mutations within the protein that lead to gain-of-function. And those gain-of-functions of the mutant form of the protein interfere with the normal KIF1A's protein function. So that's what we consider a dominant negative, a negative interfering in a very strong way. And in doing so, these mutant forms of the protein impact the transport synaptic vesicles and the synaptic precursors to the synapse. And so if you think about the cell body of the neuron is located very far away from the end of the neuron or along the axon, material has to be transported up and down that long process or the axon. And when you don't have properly functioning KIF1A, that cargo doesn't make it to its destination in the correct or timely way. And so our goal here fundamentally is to restore that normal process where cargo can be transported along the microtubule structures within the axon used by the KIF1A proteins. So as Dr. Chung's lab has been so involved in the KIF1A organization and on part of generating some wonderful natural history data from patients, what we do know about KIF is pretty well defined at this point. There's a lot of neurological concerns, obviously, since KIF1A is primarily expressed in the central nervous system. And those neurological concerns are typically hypotonia, spasticity. Many patients exhibit seizures, although they are often hard to report or hard to see or check clinically. There's abnormal MRIs, and there's been pretty significant data showing there's cerebellar atrophy in a majority of the patients. We've also know that vision or eye conditions develop in these patients. There's optic nerve atrophy, along with intellectual disability, peripheral neuropathy, autism, obsessive compulsive behavior, and anxiety disorders. And then in some populations, there's endocrine and kidney neurogenital disorders. Those are a little bit more or less common. And what I think is becoming very interesting and what has come to light within just the last year or so is that often people or patients with KIF1A associated disorder are misdiagnosed with other neurodevelopmental disorders such as cerebral palsy or Charcot-Marie-Tooth syndrome or Rett syndrome. So, and I think I've heard more recently too, there are quite a few patients that had these individuals that were diagnosed, inappropriate diagnosed with these other neurodevelopmental disorders. And so really the only way for us to know whether a person has KIP1A associated neurologic disorders through genetic testing and understanding where the mutations and what mutations there are. And at this point, Wendy's lab has identified, I think, close to 68, if not more, novel mutations within the KIP1A gene that are associated with KAND. So this is on the bottom half of this slide is the structure of KIP1A. And so you have a motor domain, you have smaller domains that lead to a more long linear aspect of the protein. And then you have this plex pH or plex homology domain here at the right side, or what we call the N terminus or C terminus of the protein. It's a large protein. It's over 1,700 amino acids long. And that comes into play in a minute when I explain our modalities and potentially how we can manipulate or go after repairing this gene. But it is a very long protein. So if you think of every amino acid being a pearl on a string of pearls, there's 1,700 of those that are required just to make up the KIP1A protein. Now, what Wendy's group has identified, and others, that's the majority of the mutations are here in the motor domain. I think close to 60 of them are here just in the motor domain. And the motor domain is the part of the protein that binds to the microtubules and functionally walks down the structure to move the cargo to its destination. The other end of the protein here is involved in cargo binding. But we know it's KIP1A is a family, one of a family of kinesin proteins. It is neuronal specific, as I said earlier, and it is responsible for the anterograde. And what anterograde means is moving away from the cell body. Retrograde means moving towards the cell body. So sometimes neurons pick things up, move them back. Whereas in the anterograde, these are important, moving from the cell body where a lot of proteins are manufactured, things are organized, they need to be moved towards their target. So this is the structure, but really the majority of the people here, people we know, have mutations here in the motor domain. And so that's the focus of a lot of our work. So what is our approach for KAND therapeutics? There's a lot of different things we can consider here. And so I'll go through these a little bit, and then I'll tell you what we're doing specifically right now to come up with these therapeutics. So the first thing we can do is knock down the defective allele. So remember, we each get a copy from our parents, one from our father, one from our mother. The defective allele is typically for KIP1A happens de novo, meaning it's not inherited necessarily, but happens during development. And that de novo allele can happen either in the paternal or the father's copy or the mother's copy. But what we can do is through using what we like to call RNAi or interfering RNAs, which are small pieces of RNA, which are oligonucleotides, that can go in and find that defective allele, bind to it, and cause the cell to degrade it and get rid of it. And in doing so, that allows the wild type or the normal KIP1A protein to function properly in the cell. So that's getting rid of the problem at the level of the genetic signal. The other opportunity we have is to disable the mutant protein. So once that mutant protein is made, it's in there, it's associating with the wild type protein and binding to the microtubules and preventing normal traffic along the microtubules. Using some technologies, we could go in, bind to that mutant form of the protein, and either remove it from the microtubules, promote its degradation or loss, so that the protein sends it off to the trash compactor and gets rid of it, or prevent it from ever getting to the microtubule. So that's disabling it, and I'll talk a little bit about what our approach is there. There is a potential to replace the defective gene, but like I was saying earlier, the size of this protein makes it a little bit challenging to go in from a technology standpoint to go in and replace it. So as these technologies develop, and that's more related to how big cargo can be put into gene therapy packages, at some point we'll be able to do that, but right now that's not the most ideal option for us. And then as many of you have been hearing lately, it's been very common or at least in the press, and actually right now in the clinic, we're starting to see this happen, is people using the technology called CRISPR to go in and edit mutations. That's a burgeoning technology. It's just coming up. The ability to use that safely is still being evaluated, but that is a possibility for us to use. And through this whole process of identifying novel therapeutics, we're going to also be very cognizant of understanding the global cellular pathways affected by KIF1A variation. So what I'm talking about above was very specific to each mutation, but really we also want to understand what's happening in a cell in a global fashion so that we can identify potential therapy that would affect anybody with KAND. And really that has a lot to do with understanding how KIF1A mutations change fundamentally the cell biology of the cell. And there's a lot of things that happen in there. When you change one little pathway, it can cause multiple other pathways to change. So there is an opportunity potentially to have a more holistic or global therapeutic for KAND. So our approach right now and what we're working on with Wendy are to do two things. First, on the top in the blue is work on mutant neutralization. So like I was saying, take the mutant protein out of the equation so that it can't interfere with the wild type protein and cause a backlog on the tracks of the microtubules. And so we're using aptamers as a technology to do that. Aptamers are, again, an oligonucleotide or an RNA-based therapeutic. And the reason we're using or considering them is just there's a lot of advantageous attributes to aptamers, meaning they're easy to make, they're easy to identify, they're quicker potentially to manufacture, they have some good attributes for delivery and safety. So we're taking that approach. We've been through a library screen, we've identified what we think are some aptamers that can bind to specific mutant forms of KIF, and we're hoping in the next couple months to understand whether those aptamers actually can disrupt function of the mutant protein. So that's where we're at here at biological testing. We still have some in vitro validation, and lead identification is getting there. We want to do a couple more iterations, and then we have to do this for other variations that we haven't tested. So we're at a good point here. We're getting some proof of concept, and hopefully then, like I said, in the next couple months, we'll understand more about how well this technology might work. As far as the KIF1A mutant knockdown, again, and this is an RNA gene targeting, or what you guys may have heard as ASOs or shRNAs. Fundamentally, what they do is they find that mutant sequence, they bind to it, and through a normal process that happens in the cell, that signal gets degraded. We've been, again, through a couple screening rounds for RNAis against some mutants forms of KIF1A, and have found some that specifically can knock down the mutant, but don't have a dramatic effect on the wild type, meaning the wild type should still be expressed and still function. So we're, again, in the process of in vitro validating those. So in the dish, making sure that they work the way we expect them. Hopefully, again, we'll have a lead that we can move forward and turn into a drug product. And drug product, most likely in this case, will be viral delivery, so AAV, and in a way that we can do this safely. AAV technology or adeno-associated virus technology has been used for some time now. There are several approved therapies for neurological conditions with AAV9, and we think this could be a way of being able to get in and treat people with KAND and do it safely and have a very long-lasting effect. So this is what we're doing now, and I think there's some other things that we're going to consider. We're building the tools. Wendy's lab is terrific. She has an amazing team that has really enabled all this activity. We have to move it forward. We're trying to move it faster, and obviously in moving it towards a drug product, meaning something we can develop and start testing in our preclinical models so that we can get to the clinic as soon as possible. And with the natural history data that Wendy has generated with you and your community, it's going to make getting into the clinic much easier, we hope. So I want to thank you for including Ovid in your meeting today. We're very committed to your organization. We are in it to win it. I think it's the best way to say it. And we're going to be standing by you guys for a while to come here. So thank you. Have a great day.